All right. So my name's Robert. I'm president of Tucson Herpetological Society. Some preliminaries about Zoom. If you have any questions throughout the presentation, please type them into the chat and I will get to those for question and answer. If you have if you want to ask a question with your voice, there's a raise hand button that you can use, or simply just wait until the end of the presentation to ask a question. Write down your questions or something like that. We're broadcasting, I'm broadcasting. Let's see if this will, from the Desert Laboratory on Tumamak Hill, or Chemamagduag, which to the Aata means Horned Lizard Mountain. And indeed, we sit here in the Sonoran Desert, homeland of many indigenous groups, but especially the A'atam, the Yoeme, aka Yaqui, the Hopi, and many others. And it's important to recognize those people. A bit of a sad note, but Don Mall, who is a member of our society and a world-renowned expert on river turtles, passed away recently. And our sincerest condolences go out to his brother Ed, who is also a THS member and lives here in Tucson, and all of their friends and family. You know, this is the last meeting of 2021, and it's really important for me to thank the board that has helped me learn to be a president. These folks served in 2021, or are serving in 2021, and I have to thank them for all of their support and their participation. Let me let some folks in here. Patrick Brown has helped in many ways, but especially helping manage our website. So if you have any issues with the website or have any questions about the website, please contact me and I'll get you to Patrick. I have to thank Howard Clark, who has been an excellent editor for our newsletter journal. We continue to produce really great stuff, and hopefully increasing our Spanish language articles as well to enhance our internationality as herpetologists. And Maggie Fusari, our treasurer, who has very patiently helped keep us on track with our desires to help fund important research. So with that, I will hand it over to Maggie, who will give us a little bit of a report or update about all of the financial and monetary cooperation that we've done with other organizations and how we are maintaining our treasury. So give me 1 second.
Okay, here I go. Whoops.
1 second. I'd like to say, if you're not muted, please mute. Oh, actually, I will mute you all. And then Maggie, you'll have to unmute yourself. Or I can, let's see. Yeah, you'll have to unmute yourself.
On the top, there it is. Got it. Okay. Well, part of this is obligatory because this is our annual business meeting and there should be a treasurer's report. But I'm going to be pretty brief with that and talk mostly about what we're asking you for and what we're planning to do. Bottom line on our treasury is we're in pretty good shape. We hold over $40,000, but $10,000 of that are held solely for the Mexican tortoise projects that we shepherd. And our speaker today, Taylor Edwards, has been involved in those and is willing to add some questions about what those are all about at the end of his talk. Our income has been healthy, mostly due to memberships and donations. And I'm going to say several times that Robert is very much responsible for supporting our donation income and keeping us alive and ready for the future. Our expenses this year were a little bit down because, of course, we haven't been meeting in person. We haven't been doing field trips, all of these things that we're hoping to bring back. But as a treasurer, I'm always happy when our expenses are less than our income. Most of these expenses have to do with fixed costs like insurance, post office, newsletter, website fees, etc. But you're going to hear later about the fact that we've given quite a few grants away this year, and we're extremely happy about that. Whoops, there we go. Now, it's the time of year when some of you think about donations, and I'd like to ask you to think about the THS. Our priorities, in case you want to target your donation for 2022, are the Low Grant Fund. Of course, we want to give more grants, and the Jarcho Award Fund. And Robert's going to tell you more about both of those very, very soon. I have a second idea, and that has to do with the fact that the holidays are coming and we have merchandise for sale. I don't know if you're set up so that you can see my t-shirt with our wonderful Gila monster on it. And I don't know how many of you realize or remember that we owe this beautiful graphic to Paul Miroka. And I put this green on top of it to show you that it's unique to us. This Gila monster carries our name. Woo! And we got shirts. You want a shirt? We got colors, styles, sizes, you name it, you can have it. And if you don't want to give shirts, how about something like this? Risha has very carefully brought up our store on our website, and you could have these and many other items with our wonderful little Gila monster in time for Christmas. So if you would, please think of a donation or a purchase. But now I'd like to show you the thing that we're most happy about. The grants we gave out this year. We gave out 6 wonderful grants to some wonderful people. And we're really looking forward to where we're going to go with this and other things next year. So at this point, if you have any questions for me, put them in the chat and I'll check that. And I'm going to stop my share and turn this back over to Robert. As soon as I can find my button, there it is. Okay.
All right, thank you, Maggie. Let's see here. All right, before I forget, if you are a member of the THS, you should have received an email on voting instructions for our 2022 slate of officers. And I encourage you to please go to your email during this presentation, look up THS elections in the subject line, And there are very clear instructions about how to vote for the slate of officers with links and everything. It's very simple. You vote yes or no. You type your name in and vote yes or no and hit submit. And by the end of the meeting, we'll know if you guys approve or of the slate for 2022. Moving on, I have to thank or acknowledge our partner. These are some of our partner organizations that we have extended exchange membership to, or have helped or have helped us in various projects relating to amphibians and reptiles and conservation. So check this out. Now we'll learn about a little bit about the Mexican Tortoise Project or some aspect of it later on. Fauno is an organization in Baja California. Turtle Conservancy, Taylor will probably mention a little bit. Yes, so this year we awarded $2,460.25 for tracking desert tortoises, for looking at thermoregulatory behavior or thermal biology of side-blotched lizards, and Gila monster health study looking at their gut biome. And most recently, we, under special circumstances provided in our Charles Lowe Fund, we provided some funds for Tom Van Devender and company, other local THS members who are looking at the natural history of Ditmar's horned lizard. And for those of you who may not be aware, this lizard was a mystery for many, many years. It was only known from 3 specimens collected before the turn of the century, before the turn of the 20th century. And people like Wade Sherbrooke and Vince Roth finally were able to determine the source of these lizards because no one knew where they even came from. So here in this photo, you have where you have Wade Sherbrooke, Dale Turner, Tom Van Devender, Steve Hale, lots of great herpetologists who have found a really robust population of these elusive lizards and are starting to photograph them and conduct basic observations and data collection. This photo was taken by board member Ross Maynard, who's an excellent photographer. But this lizard was virtually unknown for many, many years, only from a very small handful of specimens. And through the work of Tom Van Devender and Anna Lilia and his expedition and their expeditions, they've expanded the known localities from just a handful to over 20 or so. The co-PI is a college student in Cananea, Sonora, pictured with Wade Sherbrooke. He is studying these horned lizards which occur on his family's ranch. And he's very, very excited to be doing this work. Here's Dale Turner and Chip Hedcock measuring a snake. Here's another student named Nora who is holding a Ditmar's horned lizard. And this is a great image of, excuse me, a second, of Abel Salazar, the student from Cananea with his teacher, Guillermo Molina. They're the people on the ground radio tracking these lizards and taking all this wonderful data. And here's a really cute horned lizard on a scale. That's a full-grown female, and they're very unusual looking lizards. There's some close-ups of some individuals, and Jose Abel radio tracking them. part of the funding for this that we gave was raised through the generous proceeds of Heliotrope Metalworks. Spring Winders made some special herp designs, herp couture, as I call it. She carved this tortoise just from reference photos and cast it. And she also cast a horn, Rhinosoma solari skull that she found and turned it into jewelry. So as of today, they've told me that they've raised $926 for the THS. So we thank them very, very much for their generosity. The Philip C. Rosen Fund is formed, as you know. Recently, Mary Jensen and Cecil Schwalbe provided some money for that as well. This ensures that Phil's notes and manuscripts don't go to waste, don't get lost. The Sonoran Desert Toad Fund has raised over $12,000 with a little bit more coming in. This is to begin to monitor the species in light of the demand for psychedelic, for the psychedelic market. And we have Cream Design and Print, as well as Hamilton Morris, to thank for donating proceeds of their merchandise to help this cause. These are 3 projects which we have funded this year for understanding this species population in Arizona and Sonora. Risha Kota has been managing our storefront from our website. I have to thank her very much for that, and also for this wonderful Photoshop job of our local models sporting our merchandise. So thank you very much, Risha. I have to put a pitch in for these wonderful books, which have been available for a while now, but they're relevant to us because the authors are THS members and really great herpetologists. So check those books out. We hope in the next year to help sponsor or fund the follow-up book to Snakes of Arizona, which will be, well, follow-up books, 2 of them, one on the turtles and amphibians and the other on the lizards. So another reason to join the THS, donate, buy some merchandise. This will all help us, help the THS lift up the herpetological community in some way. And finally, it's my pleasure to tell you that Christina Jones is the recipient of the 2021 JARCO Conservation Award. And for those of you who may not know what this award is about, the THS established this award in 1992 to honor individuals or organizations for their service to the conservation of the amphibians and reptiles of the deserts of North America. The award is named for James Jarko of Tucson for his dedication and contributions to the conservation of amphibians and reptiles for more than 20 years. So yes, Dr. Jarko is a well-regarded veterinarian, but has done so much for herp conservation, but with very little recognition. And there are lots of herpetologists like this out there. Christina Jones is an incredible scientist. She is an early, I guess, mentor of mine. I've known her since I was in middle school and drove around the desert with her herping. She recently started working for the U.S. Fish and Wildlife Service, but holds numerous positions with herp organizations, especially in the turtle world. So congratulations, Christina. And we hope to celebrate this next year, perhaps, with a nice event. So Well, speaking of JARCO Conservation Award recipients, Taylor Edwards, who is our presenter tonight, received the JCA Award in 2012. Tonight, he's going to talk about 20 years of genetic research on a Sonoran Desert icon, the desert tortoise. I've also known Taylor since I was in high school and middle school. Is very formative personality, and glad to know him. He's a really wonderful person. Taylor is a conservation geneticist whose primary research focus for the last 20 years has been the desert tortoise. He was instrumental in revising the taxonomy of this group into what are now 3 independent species, including recently described Good's thorn scrub tortoise, Gopherus ebgoodii. That was in 2016. He first moved to Tucson in 1992, worked at the Desert Museum. This was his first intro to the region, and he's been a passionate desert rat ever since. He's currently an associate staff scientist at the U of A Genetics Corps, where he's been involved with a variety of genetic projects from fish to plants to humans, including helping to oversee the public testing for National Geographic and IBM's The Genographic Project, which I was also a lab helper in at that time. He has his Bachelor of Arts in Zoology from the University of California, Santa Barbara, and both master's and doctorate degrees in wildlife fisheries conservation and management from the School of Natural Resources and the Environment at the University of Arizona. He's a member of the advisory board of the Turtle Conservancy, scientific advisor for the Turner Foundation for the Bolson Tortoise Captive Breeding and Repatriation Project, and past president of the THS. He also works with Nat Geo and Lindblad Expeditions as a National Geographic expert, and has accompanied programs to US national parks, Costa Rica, Panama, Nicaragua, Peru, Ecuador, and the Galapagos Islands. So it's a bit lengthy, but I think well-deserved. I will stop sharing my screen and hand it over to Taylor.
Thank you so much, Robert, and welcome everyone. Thank you very much for being here. It's nice to participate with THS right now, even though we're still on Zoom, but see some familiar names out there and things like that. I'm going to be turning on my slides, and I hope that I can see the chat in case people need to interrupt with a question or something like that. But if I don't catch it while I'm presenting, then please save your questions for the end. I'm sure we'll have some time. Now let's see here. I think I'm going to share my screen. All right. Robert, is that?
There we go. You're good to go.
Okay, you can see my slide.
Correct. Yes.
Okay, good. I need to try a couple other things here real quickly. Hold on. Okay, yeah, so I'm not sure that I can see the chat while I'm doing this. So
I'll look out for you, Taylor.
Okay, thank you very much. All right, so I was asked to sort of fill in this presentation, and I actually don't have new material to present to the THS. I've presented over the 20 years I've been working with tortoises numerous times. So I'm going to kind of go through that journey, but kind of that's a personal journey of me entering the world of genetics. Some of the advances in technology that I've experienced while working with tortoises during this time, and how it's kind of opened up the questions that I've been able to ask, and how tortoises just seem to become an amazing subject to continue to ask questions and address conservation issues, as well as sort of general issues and questions about evolutionary biology. Now, I actually still consider myself a field biologist, although I've been doing genetics in a lab now for longer than I spent time in the field. But I truly did start as a field biologist, lived out of my truck for 9 months studying Mexican spotted owls, Got my start in herpetology, working with Don Swan and Matt Good, doing herp surveys and radiotelemetry studies. And that's about the time I was working to try to get into grad school with Cecil Schwabe at the University of Arizona. And so I had been doing these radiotelemetry studies. And one of the questions that came up was the relationship between tortoise populations in the Sonoran Desert. Tortoises occur in these rocky foothills, but they're scattered throughout like little islands throughout the desert landscape. And some of those populations are very small. So the question is, how often do they move between those populations? And for a long-lived species like a desert tortoise, you're not necessarily going to be able to truly answer that just with radiotelemetry in a human lifetime. So genetics was a way to sort of answer that question. I, with help of Don and Matt, wrote a heritage grant, received funding to do genetics, and it was actually my first time entering the lab outside of like lab projects in my undergraduate degree. But really didn't have that much knowledge about genetics to begin with. That's when I first entered the University of Arizona Genetics Core, which I'm now a manager of their clinical laboratory. At the time, I was just a student using some bench space and some of the tools that they had available. At that time, they didn't have geneticists in the School of Natural Resources or working with wildlife. And so I had to find a home over there. Back then, it was called the LMSC instead of the UAGC. And this was around 1999 or 2000. And what I was sort of tasked with was developing some molecular markers called microsatellites that we could study population genetics in desert tortoises. There had been studies with mitochondrial DNA. Mitochondria is an amazing molecular marker for studying species, looking at population and evolutionary differences. But relative to microsatellites, it's a slower mutating marker. Microsatellites mutate relatively quickly, so you can see changes across multiple, like a few generations. And they also are inherited by both the father and the mother, where mitochondria is only inherited by the mother. So you only have one direction that you're looking for your information in. Think of two different decks of cards with different backs on them. And if you shuffle them together, you could kind of think of that as your DNA profile with your mom and your dad mixing, right? And then you can take a look and see that 50% came from mom or 50% came from dad. The case with mitochondria, your card with the Queen of Hearts is the mitochondria, and the back of that card is always the same as it passes on from parent to offspring. And you're missing all that other diversity in the other sets of cards that have contributed to that individual. So microsatellites were going to allow us to do that. I should say, having not been in a lab, the first time I was in there and I extracted my tortoise DNA that I had collected a blood sample in the field to use, I didn't know any better. And I'm like a field biologist. I'm extracting the DNA. And at that time, I was precipitating it out of a solution with some isopropanol. And you can see this actual spool form as you do that in your test tube. And it looks like a little piece of cotton floating in there. And in order to clean up that DNA, you can put in a very thin glass rod and it'll stick to it. And then you can wash it in some ethanol and other things so that you can get to use it and isolate it by itself. When I did that with the glass rod, it slipped and I dropped it on the floor. And like any good field biologist, I picked it up, wiped it on my pant leg, and then kept moving on to the shock of everybody else in the lab. But over the course of 20 years, I've gotten a little bit better with my technique. The results of that study, looking at the population genetics and Sonoran Desert tortoises, was that they do exchange genetic information on a scale greater than one migrant per generation. So it is important that tortoises are able to move across these landscapes where now we've built roads and homes, and it's harder for them to do so. And that movement is actually very important, most likely, so that they can maintain those small populations in case of a decline, whether it be drought or disease or human mitigated as well. So that was great information as we were planning conservation measures for Sonoran Desert tortoises throughout the Sonoran Desert. That caught the attention, though, of course, of the Mojave Desert tortoise researchers like Dr. Christine Berry. The Mojave population, and I should mention at the time I started doing this research, there's only one species of desert tortoise, Gopherus agassizii, and it was split into two populations, a Sonoran and a Mojave population. That Mojave population was listed under the Endangered Species Act and was protected. And most of the research, I should say, had been going into that population. But fortunately, to people like Cecil Schwabe, he had lots of students starting to work on Sonoran Desert tortoises, trying to be proactive about learning them before we ended up in the same situation as the Mojave with a massive decline. So I was invited to participate in Mojave studies, and it also helped that I used to be a vet tech, and Dr. Jarko trained me how to be a tortoise phlebotomist. So I would go out in the field and was able to collect samples from the Mojave Desert, as well as Dr. Christine Berry's collection of samples she had archived over the years for disease studies. So we had a whole pool of blood samples that we could use, about 800 at that time. One of the studies we did was to re-examine the distribution of these tortoise populations so that we could help inform the units in that recovery plan. It was actually up for revision at that time, and we wanted that genetic information to be available as they revised the desert tortoise recovery plan for the Mojave population. And that idea of being able to tell differences between populations, which ones need connectivity or have more connectivity for gene flow, is really important for understanding and protecting a species across its landscape, not just in one single location. And now that I had been studying both Mojave populations and Sonoran populations, it led to doing comparisons between those two, using the microsatellite markers as well as the mitochondrial markers that we now had available to do that research. My first development of microsatellites, those microsatellite markers for Sonoran Desert tortoises, I was able to develop 7 of them in my first paper. And at this time, we have about 25 or 26 that we did out of my lab, plus other researchers have identified others. So there's now quite a few of these markers and able to do much finer scale studies of tortoises. So we were able to start looking across the 2 desert regions at these tortoises. And while it was something that many had suspected, we now had the tools to be able to split them in species. So it was fun to be a part of something I didn't set out to do when I first started studying tortoises, but now was entering the world of taxonomy. And so now we had the Gopherus agassizii from the Mojave population, and in the Sonoran population, Gopherus marafkai. So that was that split of the 2 species, which had been what we call allopatric speciation, the formation of the Colorado River 5 to 6 million years ago, separated those populations, and they evolved and adapted to their separate regions independently. And the genetic information helped show the time frame of that as well. Now, while this was going on, I was still working on tortoises. I was actually working at the lab as my full-time job because we had taken on National Geographic's Genographic project. And I oversaw the public testing that we did for the project. We processed over 500,000 samples for them. And the interesting thing was we were using microsatellites for this project to look at human population differentiation around the globe, in addition to mitochondrial differentiation. So using those same molecular tools in these human studies, I was applying them to tortoises. And that included things, these microsatellites are the same things they use in human identification, forensics, paternity testing. And so I was able to apply it to paternity testing of tortoises. We did a study in the Mojave through some of their Head Start programs where we had females where they could identify their clutch of eggs. We could collect samples from them and we're able to do genetic studies on them. And we found out that 64% of the females we studied had mated with multiple males. Females can store sperm, and that 50% of those clutches or those nests of eggs that they would lay would have multiple males contributing to the young. Really interesting to start thinking about conservation of species this way, knowing that a single female is carrying around not necessarily just her own genetic diversity, but that of multiple males that she's mated with. So when we think about things like translocation, or a single female doing one of these long migrations to a new population, she could actually be contributing more genetic diversity that way than just her own. Really fascinating to me, and nothing I ever thought we'd even be able to answer a question like that, and still until we started having these genetic tools to do so. We also did some tortoise forensics. This tortoise, the larger one in the picture, you can see some red and white and green paint on the shell. It was on a long-term monitoring plot for the Arizona Game and Fish Department in the West Silver Bell Mountains. To get to this site, it took me over an hour on a bumpy dirt road to even get to the location. Peter Woodman had been working on that site multiple seasons to track population demographics, looking for things like trends and declines or things like that, population levels. And this was a tortoise he had seen 4 years earlier with the entire shell painted like a Christmas scene, again, in a pretty remote location. And then when I was there with him, we found it again, we're able to get a blood sample. So the questions are, was this tortoise brought there from another location? Did somebody show up there and just paint it on site on some Christmas morning? Was it an escaped pet? And this is not an uncommon thing to find with tortoises. The other tortoise there was actually one we saw crossing the road in Mexico during our studies there with a fully painted shell. So applying microsatellites, these same kind of markers, we're able to do a genetic study looking at trends in captive tortoises in populations in Arizona. The tortoise in the West Silver Bells actually genetically seemed very much like the other tortoises in that region. So it did seem to be a local tortoise. But we found for captive tortoises that in Tucson, 100% of the tortoises that we looked at matched local populations, so Sonoran Desert tortoises. In Phoenix, only 68% matched what would be a local population of Sonoran Desert tortoises, with the rest being Mojave tortoises, so from across the river, and then 11% of those were hybrids. In Kingman, it was even more, and I'll get to sort of this story in a minute. Only 20% were Sonoran, with the majority being Mojave Desert tortoises. They're closer, of course, to the border with California, and there's Mojave Desert interspersed a little bit in that region as well. So this, now using these markers, really got me interested in this idea of hybridization between what are now two species, not just different populations of the same species. And so that was what I became really interested in looking at next in this region of Kingman, Arizona, where there's the Black Mountains, and it's an area where there's a ecotone. So that's two different ecosystems sort of coming together. And that's the Mojave Desert, now on the east side of the Colorado River, and the Sonoran Desert on the west side of it. And so I had samples from California in the Mojave Desert. I had samples from the heart of the Sonoran Desert, and then this area where they come together, and we see a little bit of both types. I should mention that Anne McCluckey started doing this work with mitochondrial markers and found that there were Mojave type tortoises on the east side of the river in the Black Mountains. And when her paper came out in 1999, and I was just starting my work, it always sat in the back of my mind as something that'd be great to find out more about. The work that I ended up doing on this, we were able to do time, both look at hybridization and also sort of the timing of when those Mojave tortoises crossed that river, because if they were split as a species by the river, how did they get back across? And the time frame was within human scopes of time, 2,400 years or so ago. It could have been human transport by people. They could have rafted or floated across, but the one that made the most sense looking at all of the evidence that we had was that there were geological events with movements of the Colorado River channel, sometimes spanning out, periods of drier times where tortoises were able to move, not just as one or two, but populations being isolated from one side to another, and finding habitat that they could persist in. So we did some habitat modeling. You can see the yellow is Mojave Desert vegetation. The red is Sonoran Desert vegetation. This is sort of that same region I showed you on the map originally. The triangles are contact zone sites, and where we found the Mojave tortoises were in the Mojave Desert vegetation, as we would expect, I guess, and then the Sonoran ones in the surrounding Sonoran habitat, with hybridization minimized just to those kind of ecotones, but still really maintaining their own species, not just blending together throughout that area. So that was really fun to start looking at that, and it really made me start thinking about deeper questions about the distribution of these species, how they evolved across these landscapes. It was also around this time that with collaborators, we started the Mexican Tortoise Project. While tons and tons of research had been being done, particularly in the Mojave Desert, because of them being a listed species under the Endangered Species Act, very little was known about their conservation status in Mexico. So we did a collaborative effort with Mexican colleagues, but to look at everything we had specialties in: genetics, health and disease, behavior, movement, all of those things with an incredible team. And we spent 8 to 10 years collecting samples and visiting sites down there for this project. Now, as I said, I consider myself a field biologist. I love being outside, but at this point I had been working in a lab for almost 8 years. And I do have to say there's something I didn't expect I'd find when I started working in genetics. Maybe it's because I have a short attention span, or I just like the instantaneous rewards that you can get from genetic studies. But when we got our first samples back from Mexico, and I did some mitochondrial sequencing, it looked a little something like this. And you can see on this slide different patterns showing up of the letters of the DNA sequence. I'm not going to go into too much detail, but those patterns just jumped out at me immediately. And in a matter of like a day or two, I'm looking at these samples and saying, this is something really different down there. These were samples from southern Sonora, and I knew that this was something bigger than just sort of a stepping stone model of their relatedness going down south. And that kind of question got me wanting to look more into these deeper evolutionary questions and using these genetic markers to do so. When you look across the Sonoran Desert, you can see sort of what we have in our surrounding area here in the yellow, and that's sort of Sonoran Desert scrub. But it transitions as you move south, just sort of as you're getting to Hermosillo and a little past, into this thorn scrub habitat, and then into tropical deciduous forest as you start to get into Alamos area and into Sinaloa. And these habitat types, as we showed with the Mojave and Sonoran deserts, when they're intermixed, is the tortoises sort of partition based on their adaptations to those environments. So what's going on down here? Evolutionarily, there's no giant mountain range or river that would have separated these differentiation. And we also know that these vegetation types moved over time, over the millions of years through the Pleistocene as the climates changed and during ice ages. So they weren't just in one place all the time. So that would affect the species like tortoises that are adapting to them as that's occurring. So it's sort of like a moving picture over time, and we're trying to trace that to how the species have adapted and evolved. When I look at a mitochondrial map of that, we can see the breaks and the differentiation happening right along that thorn scrub line about 5.7 million years ago. And interestingly, that's about the same time frame that we see the break across the Colorado River between the Mojave and Sonoran Desert. But this had so much more going on because of this, what we call gene flow, this ability of these tortoises to potentially intermix, that my microsatellite markers weren't going to be quite enough for me to answer this question. This is kind of what motivated me to get my doctorate at this point. Because of the new technologies I was being exposed to in the laboratory at the UAGC, at the genetics core where I worked. Now we had things like next generation sequencing, where we were doing human genomes and exomes and all of these things. And I wanted to apply some of that to tortoises. In my case, I decided to use what we call RNAseq. I was collecting fresh samples and extracting RNA from them from these different regions, Mojave, Sonoran, and the southern Sonoran in Mexico, and looking at the sequences of these express genes through the RNA. But unlike those 24, 25 microsatellites that I was using before, I was looking at over 20,000 different non-coding markers from all across the genome. That's what these kind of contates are representing. So now we had a lot more high resolution tools to do this work. And I could start to ask questions like, okay, if these are species that are splitting, did they split and completely just separate with never coming back together, what we might call an allopatric speciation model, or did they continually come into contact with each other, interbreed, and then sort of divide since that time? So what's the time frame for them being able to exchange genetic material as they were diverging? Ultimately, we found that we now had a trichotomy, sort of three different types of desert tortoises, the Mojave, the Sonoran, and what we would call the maybe Sinaloan type tortoise, which we ended up calling the thorn scrub tortoise and was defined as a new species. But very interestingly, those models of evolutionary change were the same, whether there was the river there or was simply just an ecological difference where they adapted to different habitats, that thorn scrub and tropical deciduous forest, or to the Sonoran Desert scrub around the same time. So my research and my thinking about things became so much deeper in time to answer questions that we were seeing in front of us now. And also by defining species, and this is Gophorus ivagudi, we're able to now channel resources to protect species, whereas before we considered it one giant population. We might say, oh, if some we don't save at the end of their range, that's okay. In this case, we have a new species in the southern part of the range that's actually the smallest population of the desert tortoises and in some of the most imperiled habitat in tropical deciduous forest. So that was an exciting thing to be a part of. And definitely great to be a part of a team doing such a broad study to get that kind of work done. Now, jumping into that world of what we call next generation sequencing, or this RNA-seq that I was doing, led to actually doing more genomic work and contributing the data that I had, and with collaborators at ASU. We published the desert tortoise genome, which is kind of crazy for me to think about. This was in 2017 that we published the genome. And when I first entered this sort of world of genetics, I'm going to say right around 2000, was right when President Bill Clinton and Tony Blair announced, they declared that the human genome was complete. It had started in 1990, a 10-year project to $2.7 billion, which is actually less than they anticipated to do that. Now, while I knew those were things that were happening, I never thought that that would be a part of the tools I had to work with tortoises with. And then here we were in 2017 with the desert tortoise genome to continue with. So with a genome, unlike the previous work that I had been doing, we can actually start to look at the actual genes that are adapting to these changes. As these tortoises are evolving and becoming different species, adapting to their environments, we can actually look at differentiations in genes. And similarly, how they differ from different turtle species. And now there's been an effort to get more turtle and tortoise genomes out there as well. So those are some really exciting things, again, that I never thought that I'd be a part of when I first started. And another really fun thing, seeing it about 20 years later since developing those first markers and working with those tortoises, was actually revisiting some of my earlier work with these new tools. This was a project that was done with both some new samples, but also revisiting those older 800 or so samples that we had collected from the Mojave Desert, but now using these new genomic tools, many more sites across the genome. And one of the rewarding things was to be able to see that the same patterns were showing up. My earlier work was supported with this higher resolution data in where we had sort of drawn the partitions around these populations for the recovery units in the Mojave Desert. But what also was contributing is that we could also start to see that there's functional genes that may be involved in that partitioning. The Mojave Desert's a very large, wide-ranging place, all the way, from Utah down through the southern Mojave and these creosote flats and things like that. So obviously the animals that live there need to be adapted to survive those harsh environments, whether the cold freezing winters in the north or the desolate dry areas of the south. And so certainly they have genes that are functioning with that. By partitioning, by better understanding that, again, we can better manage them as populations. It can inform translocation and those kinds of things if we want to ensure that they have the best chances for surviving. So that was a fun study to revisit. And then Greer Dolby, I've been working with their team at ASU as well, and they recently reevaluated using some of these genomic tools, some new populations in Arizona, close to the Colorado River. And you can see in this image, there's sort of a pink and a pink-purple dot in the, I'm gonna say, center bottom of the screen. Those are populations in what is Mojave-like habitat again, where they're again finding hybrids further to the south as we would have predicted, but didn't have the samples or the data support when we originally started studying hybrids. So it's really fun to see these ideas and tools that were developed continue to be used by other scientists to find out more information. So I still work at the genetics core. I'm the clinical lab manager, which has changed a lot of what I do during the day. I've actually never had it be my job to study tortoises. It's always been a passion and part of my education. I will continue to study tortoises, and I will continue to be a field biologist and be outside with them too. But for the last year and a half, I've actually been developing COVID tests and running hundreds of thousands of samples with my team at the University of Arizona. And I'm enjoying the other scientific opportunities that genetics has provided too. Through the human genetics that I've worked with, applications to precision medicine, it's pretty amazing. And though I might not have expected it to take me where it has, it's been an incredible journey with many more opportunities to come. I do have to say thank you to the many people who helped me get here, both from the lab and in the field, my mentors and colleagues. And there's many, many more that I haven't listed here, but none of this work, of course, happens just by itself. And with that, I can answer some questions, but that is sort of my quick personal journey into genetics using desert tortoises, who have become my subject and continue to spawn new questions for me as I go. So thank you very much.
Thank you, Taylor. That was great. Oh, let me mute some folks here.
going to do female tortoises carry viable sperm as long as a year or more? The answer to that is yes, and they have. We found this primarily with the Bolson tortoise, where we're doing the captive breeding for that population on the Turner ranches. We had a tortoise have a clutch with a male tortoise that we had a sample from, but that had was deceased. So that was longer than a year. And we're also seeing that with some studies. I'm continuing to do some of the paternity work with some of the Mojave Head Start populations, and Tracy Tuberville, who's been working on those as well. So we don't know how long. There's anecdotal evidence of tortoises in captivity that it continue to sire young or have young as or lay viable eggs. I feel like anecdotally up to 10 years, but I don't have actual proof for that. So that would be interesting to know how long that can go. We have what roughly are the differences in longevity of the different species and or within populations? You know, that's a good question. Most of what we know for longevity, because they live so long, is from captive tortoises, which we know they can live over, you know, up to or a little over 100 years in captivity, given the right conditions. In the wild, with our telemetry studies, they live typically less than that. 40, 50 years might be a long-lived tortoise in the wild. But we don't have comparative study information for the three species, and particularly not the thorn scrub tortoise. It's the least known, and we haven't had long-term studies to even be able to start looking at that.
I'd like to make a quick anecdote. There was a note in Journal of Herpetology that looked at some of the mortality, non-human mortality of tortoises in the wild, and there was predation from mountain lions and getting stuck in rocks, getting wedged between rocks and not being able to get out. And the reason I bring that up is because the original name for Sells, Arizona, on the A'atam Reservation, the original indigenous name for that place means tortoise stuck in the rock. And apparently from a certain angle, you can see what looks, one of the hills looks like the butt end of a tortoise stuck in the rocks.
And I've found deceased tortoises stuck that way myself. You probably have too, Robert. Yeah. So it does happen. Have we identified any ecological or physiological adaptations for the three populations, especially the thorn scrub population? At this point, just anecdotally, as far as where they are situated, that will take more work. We know a lot about Mojave and the Sonoran population in the thorn scrub and tropical deciduous forests. There have been some telemetry studies that started while we were working with them, but I'd love to see more. And this is definitely a population that could use more research. And as Maggie mentioned, we've got a research fund. And so we'd love to get people down there to do this kind of work.
Let's see. Brian wants to speak, so look him up and unmute him.
Hi, Brian.
Hey there. Thanks for that talk. It was great. I just thought it'd be easier than me trying to type this in on my little keyboard here. I'm curious, and it's obviously a very difficult and yet important question. Your sense of the population structure as the little islands of rocky slope habitat that they inhabit in the Sonoran Desert. How worried are you about the subdivision of that habitat? Meaning, like all the populations I work with here in the Phoenix area are all now isolated islands. They can't make those movements. Those are the ones that, you know, are prevented from doing so by roadways and the rest of it. How worried are you about Murawski's long-term success probability given the need for those, or the apparent the need, the fact that it's been the common situation for most of their history that they do exchange individuals?
That's a great question, Brian. And that's actually what got me into doing this genetics work, was that sort of landscape connectivity question to begin with, because I'm worried about it, and I still am. What I've realized working with these long-lived, slow to mature species is that it's not these aren't populations that are going to disappear necessarily in our lifetime, although we might see their demographics change over that time. And we do see populations get sometimes hit with drought or disease, and they're going to be slower to recover. But it's on a different scale than we're used to thinking about conservation, because we got to think hundreds of years in order to know what's going to happen to these small isolated populations in some cases. Maybe with climate change and stuff, it'll be sooner we're going to see what's going to happen to some of them, because they've undergone massive climate changes over their evolutionary history, but they had places to go, and they had populations to contribute new genetic material and adaptation from. There was a cartoon that Don Swan used to always reference, which was, this was during the Reagan era, and it was a tortoise on a rock talking to the tortoises below, and he says, "Ask yourself, are you better off now than you were 400 years ago?" And it truly is not the way we think about conservation. People make these kind of plans and changes often in very short time scales, election time scales and things like that. I'm worried about it because we're starting to plan that way, and we have great projects maintaining connectivity in areas. The Mojave Desert plan is coming together, trying to keep these regions done, but we really do need to keep thinking big and not just on small populations. Because in Tucson we've got Tumamoc Hill. I did sampling up there. There's maybe 50 tortoises up there. They're pretty much an island. They'll probably be there in 100 years, a small population of tortoises, but that's not what's going to allow the species to persist. So thanks for asking that question.
I see we've got some new visitors here. I think they might have thought that the meeting was at 7. So, but this will be recorded so you can watch it on YouTube. Taylor, I've had a question for you about one thing that I've gathered from your talk was, seems like there are many ways of interpreting what a species is, and it seems like you've been successful at explaining or delimiting or outlining species. Do you have any just sort of general reflections about do we still know what a species is? Do we have a better understanding of what a species is now than we ever have? Or
it's actually it's probably become worse because of all these darn geneticists out there giving us new tools to look deeper into our connections to things. So, the biological species concept of do they interbreed and have viable offspring, that was sort of the standard for a long time. We now, as we look across nature, we see so many different amazing types of species that we call species. And the definition that I primarily go by as an evolutionary biologist is their evolutionary potential. As you see things diverge, they may still be able to hybridize, but they also are diverging on different tracks. And that takes a long, long time. So it depends on where we take the picture of these diverging species over time. And my projection would be if things were stable, which they never are in nature, these tortoises over another 6 million years might not be able to hybridize. We also have to consider that tortoises don't have sex chromosomes. And a lot of the sexual, the drivers for a species not being able to interbreed can sometimes be on sex chromosomes because you only have one copy. It's easier to pass it through a population in a way, whereas you're always shuffling the deck. It may not take hold at the same rate of time. So that's one factor. And the other is it's probably really beneficial to sometimes be able to hybridize when you're in these ecotones and habitats where it might be beneficial to those individuals to have some adaptations. And again, this is looking at these actual genes and their selection that way. It's probably very helpful to hybridize. We look at things like wolves, which we call definitely a different species than a coyote, but they hybridize with coyotes. Sometimes we find a mitochondrial DNA. And interestingly, we also find other parts in their genome that have been contributed not just recently by coyotes, but by coyotes in the past. And when I was doing that, the genographic project, Dr. Hammer, who is the PI of the lab, was also doing lots of human genetic studies and published one of the first papers on human hybridization, Homo sapien hybridization with Neanderthals as they entered Europe, finding small bits in the genome that reflected that these two different species had hybridized. We still obviously consider us a different species from Neanderthals, but we have some actually pretty good genes we got out of that. Dentition on our teeth, more hair moving north to keep us warmer and some things like that. So the act of hybridization by itself doesn't negate two species ability to still be differentiated. That's probably a much longer answer than you wanted, but
no, that's
but yeah, that's something. Those are the kind of things. Once I started looking into those questions, I had to dive a lot deeper into.
No, that was a great answer, and you just reminded me that I saw a Science News blip that suggested or said that one of the genetic characteristics of Neanderthals in living humans might be beneficial in some way to dealing with COVID or something like that. So that was something that blipped up on my screen.
I did just, Brian asked a question in the chat about observing females passing through home ranges of multiple males when foraging post-monsoon and have mated with multiple males during those moves, which may be beneficial to her and the fitness of her offspring, certainly. And what's really interesting is the males are territorial, so they probably don't want those females to leave their area. And if any of you have ever observed them with females, they can be pretty aggressive too, sometimes blocking them in a den so the female can't get out, and some things like that. It's always been another goal with my partners, Christine Berry and Bob Murphy, to do these behavioral genetic studies, of which Dr. Berry has over 20 years of field observational data on. And so we do have some RNASeq data on all those tortoises, and I think we need to have the grad student who's going to put it all together.
Can I unmute again here? I just have, so I'm hoping that's okay. I was just a bit surprised, Tay. I agree with you completely. My tortoises are territorial, the males. The females are not. But when I got into the tortoise world, and you know I'm a real latecomer, it's just been these last 10 years. I'm in my 10th year with that population, and most of those individuals were tagged this fall. But in any case, I became convinced that they are territorial. But when I would speak about this at the Desert Tortoise meetings in the first 5 years, folks said, well, no, we don't have evidence to support either Agazii or Moravkai are truly territorial. They kept saying it's a home range, it's a home range. And so I've, minded my piece and kept on, kept that on the lowdown. But I'm utterly convinced. Now, it may be that I have a low density population and the males can, quote unquote, afford to be territorial, because some people think that high density populations like, up here, that would be the sugarloaf population that Game and Fish works with. They think their males overlap too much. What I find is that they may overlap in a year's spatial sense on a PowerPoint slide, but my males who have overlapping home ranges never overlap temporally. The only time they're together in the same space, they're fighting. And so I'm convinced that even if they don't have an exclusive territory, that the spatially is exclusive the way we think, like our chuckwallas, they have absolutely spatially discrete home ranges. And most of the lizards we think of are like that. But for my tortoises, I think that people have been misled by their finding of overlap of minimum convex polygon home ranges, and that in fact, I think they're territorial, and if people look into it. So I'm pleased to hear you say it, and I'll stop talking.
Sure. Yeah, if you've ever seen two male tortoises battle it out and flip each other over, they're fighting over something. And I don't know if we've pinpointed what that resource is specifically, whether it's dens, but it's likely has to do with females as well. So.
All right, well, I want to be mindful of people's time. So I would, before we go, it seems like everyone has voted unanimously for the 2022 slate of officers. So thank you very much for voting. And one thing that I forgot to mention, maybe perhaps early before the meeting started, was that we have reserved the U of A Hari Lecture Hall for member meetings, which will be a hybrid between Zoom and in person next year. So that's great. And coming up on December 5th, we're going to have we're going to participate in a fundraiser for equitable science education in public schools. So we'll have a THS table there and coloring books and crayons to play with. There'll be a trivia game and all of that. So yeah, look out for that. We hope to also continue field trips or pick back up with field trips next year. So anyhow, thanks so much, Taylor.
Thank you, Robert, and thank you everybody for attending. I appreciate it.
All right, I'm going to now end the meeting for everyone.